GXP2V. I want you to just hold that acronym in your head for a second. GXP2V. It sounds kind of innocuous, doesn't it? Like, I don't know, a serial number on the back of your Wi-Fi router or maybe a license plate. It's boring, sterile.
Very clinical.
Yeah, exactly. But in the last few weeks, that specific string of characters has become the subject of some of the most urgent and, frankly, terrified conversations I've ever seen among virologists and biosafety experts.
Absolutely.
Because GXP2V isn't a piece of tech, it is a mutant coronavirus. And the source material we are doing a deep dive into today, well, it reads less like a scientific paper and a lot more like the opening chapter of a thriller you read on the beach and then, can't sleep afterward.
It really does. And you're right, boring is exactly how these things usually start. You see a preprint paper pop up on a server, in this case it was Bio4Cause, and it looks completely standard. Just another data point.
Right, just science happening.
Exactly.
Yeah.
But then you look at the authors, specifically the Beijing Advanced Information Center and other institutes, including the 5th Medical Center of the PLA General Hospital.
And that is the Chinese military.
Yes, it is.
Yeah.
And then you look at the results table in the paper, and you realize this is something very, very different.
Right. And before we get into the nightmare fuel, and trust me, there is plenty of that coming for you today, let's set the table properly. We are looking at a specific preprint paper that analyzes a pangolin coronavirus. But our mission today isn't just to recap a scary story to spook you.
No, not at all.
We need to understand why this specific experiment represents a massive critical shift in virology. We need to look at the technical gap between studying nature and arguably creating risk.
That is the core tension we are going to explore. It's the dual-use dilemma that basically haunts modern biology.
Right.
We have this drive to understand pathogens so we can fight them, right?
Yeah.
To build vaccines, to predict pandemics. But in the process of understanding them, we often have to manipulate them. We amplify them, test them. And the question this paper raises is, at what point does the testing itself create the existential risk we're trying to avoid?
The source actually calls it a mutual arms race, which is a chilling phrase to hear in a biology context. So let's look at the weapon in question. The study revolves around a virus tested on mice. But I want to be clear for everyone listening, these weren't just standard lab mice you'd find in a pet store.
No, and that's a crucial distinction. If you take a human targeting virus and spray it up the nose of a regular field mouse, usually nothing happens.
Really, nothing.
Right, because their biology is just too different. Their cells don't have the right handshake for the virus. So for this study, they used humanized mice, specifically transgenic mice.
Okay, let's unpack transgenic. It sounds very sci-fi, but what does it actually mean for the animal?
It means the mouse's genome has been edited. In this specific case, they inserted a human gene into the mouse's DNA. And that gene is for the ACE2 receptor.
ACE2. I feel like we all became amateur virologists back in 2020, so that definitely rings a bell. That's the doorway for COVID, isn't it?
Precisely. Think of a cell like a house. It's locked. A virus is a burglar, and it needs a key to get in. For SARS-CoV-2 and for this pangolin virus we're discussing, the key is the spike protein, and the lock on the cell is the ACE2 receptor.
Okay, I'm with you.
So a normal mouse has a mouse-shaped lock. The virus's key doesn't fit. The burglar tries the handle, it's locked, and the mouse lives.
So the virus knocks, nobody answers, the mouse walks away fine?
Exactly. But in these transgenic mice, the researchers essentially swapped out the locks. They engineered the mice to express the human version of the ACE2 receptor.
Wow.
Yeah. So now, biologically speaking, the mouse's cells look human to the virus. It allows researchers to simulate a human infection model without, well, using actual humans.
Okay, so we've got mice that are effectively chemically disguised as people. They are proxies. Now, let's talk about the intruder. The virus itself. You mentioned it's a pangolin coronavirus. Does that mean they went into a cave, swabbed a pangolin, and this is exactly what they found?
No, and this is where the story gets really complicated and where the arms race aspect comes in. The virus is derived from a sample taken from a pangolin years ago, a strain called PCOV-GD01. But the virus they used in this study, GXP2V, is what's called a cell culture adapted mutant.
Cell culture adapted mutant. That implies it changed in the lab, not out in the wild?
It did. When you grow a virus in a petri dish, culturing it in VRO cells, which are monkey kidney cells commonly used in labs, the virus evolves. It adapts to that very specific environment. It's just trying to survive in the dish.
Right.
In this specific case, the researchers noticed that the virus had spontaneously mutated. It had changed its genetic makeup on its own.
So nature basically rolled the dice in the dish.
In a way, yes. But here is the major pivot point. Instead of just noting it and moving on, they isolated that mutant. They said, hey, this looks interesting, and then they cloned it.
Wait, they cloned it?
Yes. They used the tools of the lab to mass produce it. They generated a massive stock, trillions of copies. So GXPTV isn't something you'd stumble across in a jungle. It's a specific lab-amplified version of a mutated virus. They wanted to see, what can this specific mutant do?
So, just so I have this straight, nature provided the clay, but the lab shaped the brick and baked it.
That's a good way to put it.
They created a pure sample of this mutant to test its pathogenicity, its ability to cause disease. And when they introduced this clone to the humanized mice, what exactly happened?
Complete devastation. The headline figure that has everyone talking is the mortality rate. It wasn't 40%, which would be high. It wasn't 90%, which would be catastrophic.
It was 100%.
100%. 4 out of 4 mice in the infected group. Every single one died within 7 to 8 days. And the paper notes something really interesting here.
What's that?
The researchers themselves seem surprised by this. Usually in these types of spillover studies, you might see the mice get sick, lose some weight, maybe a few die. But 100% kill rate is incredibly rare for a coronavirus study of this nature. It wiped them out completely.
That is incredibly stark. It's a clean sweep. But I want to dig into the how. Because when we hear fatal coronavirus, we all have a script in our heads from the pandemic. We think lungs, we think pneumonia, ventilators. Is that what killed these mouths?
That is the most terrifying part of this entire deep dive. No,
That's it.
it wasn't the lungs.
But it's a respiratory virus. You inhale it.
You do, and initially the study shows high viral loads in the trachea and the lungs. That's standard. But unlike COVID-19, where the immune system often freaks out and floods the lungs with fluid, that cytokine storm we heard so much about, these mice didn't die of respiratory failure. Their lungs, relatively speaking, held up.
So the virus didn't just stay in the lungs.
It migrated. It broke out of the respiratory system and traveled. The cause of death was a massive late-stage brain infection.
It went straight to the brain.
It invaded the central nervous system. The technical term is encephalitis. The virus effectively ate away at the brain tissue. And the physical symptoms described in the paper are, well, gruesome is the only word for it.
Yeah, I read the source material, and describing it as nightmare fuel is putting it lightly. It details the last days of these mice. It lists things like rapid weight loss, obviously, a hunched posture. But there were 2 things that really stood out to me. One was piloerection.
Which is the medical term for hair standing on end.
Impressive.
It's a sign of extreme physiological distress and temperature dysregulation. The animal's autonomic nervous system is going haywire.
Right, and the other symptom, and I honestly still can't believe this is printed in a scientific paper, was the eyes.
Yes. The white eyes.
The paper explicitly says their eyes turned white. That sounds like a detail from zombie fiction. What is biologically happening there?
It's likely a combination of severe inflammation, cataracts, and viral invasion of the ocular nerves.
Yeah.
But visually, it signals a complete systemic takeover. The virus wasn't just in the breathing tubes. It was in the eyes, the brain, the nerves. It was everywhere.
And this brings us back to those locks we talked about, the ACE2 receptors. We know they are in the lungs. Why did the virus go to the brain?
Because humans have ACE2 receptors in the brain too. This is something the study highlights and something that doesn't get talked about enough. The human brain expresses ACE2.
Where specifically? Because if it's just in some non-critical area, maybe it's manageable.
Unfortunately, it's in highly critical areas. The substantia nigra, which handles movement, the cortex, the hypothalamus, which regulates your body temperature, and most critically, the brainstem.
Which tells your body to breathe.
Exactly. If a virus targets the brainstem, it simply doesn't matter how clear your lungs are. If the signal to breathe stops, you stop.
So the implication here, the reason you need to care about this, is that if a virus like GXP2V were to spill over into humans, we might not be looking at a bad flu. We could theoretically be looking at a 100% lethal brain infection.
That is the theoretical worst-case scenario the study presents. It suggests a mechanism of death that totally bypasses our usual treatments for respiratory viruses. Ventilators don't help if the brain is compromised. Antivirals that don't cross the blood-brain barrier wouldn't help. It changes the game entirely.
That is a heavy realization. We have a virus that kills all of its subjects by essentially attacking the control center. Now, if I were running a lab and I held a vial of this stuff, this cloned, mutant, brain-targeting virus, I'd want to be in the deepest, most secure bunker on Earth. I'm talking spacesuits, multiple airlocks, thumbprint scanners.
You're describing biosafety level 4 or BSL-4. That's the hot zone standard. That's where we keep Ebola and smallpox.
Right. So looking at the study, was this done at BSL-4?
No. And this is where the scientific community is really raising the alarm and where the ethics of safety come into play. The consensus among experts reviewing this paper, people like Justin Kinney from Cold Spring Harbor Laboratory, is that this was likely conducted at BSL-2.
BSL-2. Okay, give us a reference point. What usually happens in a BSL-2 lab?
Routine blood work, testing for salmonella, maybe some basic staff research. BSL-2 is what you find in a standard university teaching lab or a hospital diagnostic room.
So we're talking dentist office safety levels.
That's step up from a dentist, but not by much in the grand scheme of things. In BSL-2, you're often working on an open bench. You wear a lab coat and gloves, maybe a face shield, but you aren't in a pressurized suit. You aren't necessarily working in a negative pressure room that prevents air from escaping.
But we just established this is a respiratory virus. It travels through the air.
Correct. If you are working with a potential pandemic pathogen that is airborne, and you are doing it on an open bench, one splash, one drop vial, one sneeze from a mouse, and that virus is in the room.
And if it's in the room, it's in the researcher. And then the researcher finishes their shift and gets on the subway.
That is the nightmare scenario. Critics like Justin Goodman from the White Coat Waste Project have been very vocal about this specific study. He called it a recipe for disaster. You're handling a pathogen with unknown human transmissibility and known total lethality in human proxies, using safety protocols designed for food poisoning.
Why? Is it cost? Is it arrogance? Why would they do this at BSL-2?
It's often convenience and speed. BSL-4 labs are rare, expensive, and slow to work in. There are only a handful in the world. BSL-2 is easy, and often regulations lag behind the science. Until a virus is proven to be a human killer, it might not be classified as requiring high security. But in this case, the experiment proved it was a killer, yet the safety levels presumably remain static.
It feels like checking for a gas leak with a lighter. You find the leak, but then boom. Now, we have to talk about the elephant in the room, the authors. You mentioned the PLA General Hospital, the Chinese military.
Yes. The 5th Medical Center of the PLA General Hospital is a listed affiliation for one of the key authors.
Does that mean this is a bioweapon? I know that's where the internet's mind goes immediately.
The source material is careful here, and we should be too. Most experts who analyze the methodology say this doesn't look like a weapons program. It looks like academic research. But the military connection adds a layer of opacity and, paradoxically, a layer of surprise.
Surprise. How so?
Because usually, military research is black-boxed. It's secret. You don't put it on a public preprint server. But this paper, it was incredibly transparent. They detailed everything, the cloning, the lethality, the brain infection. The source analysis suggests this might have been a mistake.
You mean they didn't mean to publish it?
Or they didn't realize how the world would react. It might have slipped through the net, as the source puts it. It's possible the researchers thought, look at this amazing discovery we made about viral pathology, without realizing they were essentially broadcasting, look at this doomsday bug we just brewed up in a medium security kitchen.
Slipped through the net. That's a comforting phrase when discussing a brain-eating virus. But let's zoom out. You mentioned the arms race earlier. This isn't just about one lab in Beijing, is it? We can't just point fingers there.
No, not at all. This is a global issue. The logic of gain-of-function research, or whatever term you want to use for enhancing pathogens, is driven by a need to be prepared. The argument is, nature creates threats. We need to anticipate them, so we simulate them in the lab to test vaccines and treatments.
We have to build the monster to test the shield.
Exactly. But the counter-argument, and it's getting louder with studies like this, is that by building the monster, you are creating a risk that didn't exist before. Nature didn't create GXP2V. The lab did.
And if the lab leaks, you've caused the exact pandemic you were trying to prevent.
That's the irony. The researchers state in the paper that they wanted to assess the spillover risk to humans. But by creating a clone that is entirely lethal to humanized mice, they actively manufactured that risk. As one observer in the source notes, I'd rather not die than know about some obscure aspect of viral pathophysiology.
Amen to that. I'll take ignorance and survival over knowledge and extinction any day. But if this is an arms race, it implies there are two sides. If China's doing this, is the U.S. doing it?
That's the complicated part. Scientific research is international. Funding flows across borders. Now, it's important to note the source explicitly says there is no evidence this specific study used U.S. funding.
Okay, that's an important disclaimer to have out there.
It is. But the culture of virology is global. If leading labs in one country are pushing the boundaries of what's possible, cloning mutants, Testing on humanized mice labs in other countries feel pressure to keep up. It becomes a self-perpetuating cycle of risk.
It sounds like nuclear proliferation.
It's very similar. Except, as the source points out, we have treaties for nukes. We have the IAEA. We have inspectors. If a country starts enriching uranium, the world knows. But biology, you can do this in a room the size of a Starbucks.
And the bomb can replicate itself.
And it can walk out the door in a researcher's respiratory tract.
That is the terrifying distinction. A nuke sits in a silo until you launch it. A virus is an active participant. It wants to spread. It evolves. And in this case, it evolved in the dish, and the researchers said, hey, let's make 1 trillion copies of that.
It highlights that we're dealing with biological systems that are unpredictable.
Sure.
The researchers admitted they were surprised by the lethality. That admission is key. It proves they didn't know what would happen. And if they didn't know that, what else don't they know?
What else is in the freezer?
Exactly.
We've covered a lot of ground today, and frankly, most of it is terrifying. Let's try to distill this. If you are listening to this and need to take 3 things away from this deep dive into the GXP2V protocol, what are they?
First and foremost is the mechanism. We aren't just looking at another pneumonia virus. We are looking at a study that demonstrated 100% mortality in humanized mice via severe brain infection. It bypassed the lungs and attacked the central nervous system. That is a capability we cannot ignore.
Check. A brain-targeting, lethal pathogen. What's number 2?
Number 2 is the safety gap. This research on a potential pandemic pathogen was likely conducted at BSL-2, a safety level designed for moderate risks, not lethal airborne viruses. It highlights a massive disconnect between the danger of the organism and the security of the lab.
And number 3?
The origin. This wasn't a wild virus found in a cave. It was a mutant that occurred in a lab culture, which was then cloned and amplified by researchers. It underscores that human experimentation is actively creating new, high-risk viral strains that do not naturally exist.
And there's one final detail you mentioned to me during the break about the paper itself. It's a preprint.
Yes, it hasn't been peer-reviewed yet. And given the backlash, the controversy, and the military links, there's a very real chance this paper might just disappear.
Disappear entirely.
It might be withdrawn. It might be buried. If the transparency was indeed a mistake, we might never see the final version published in a major journal. We might have gotten a glimpse behind the curtain right before they pulled it shut.
That's a chilling thought. The data vanishes, but the virus remains in the freezer.
And the knowledge remains in the heads of the researchers. You can retract a paper, but you can't unlearn how to clone a mutant.
Which leaves us with a really profound question. We live in a world where we accept that nuclear risks are managed by treaties, red phones, and intense oversight. We worry about chemical weapons, but when it comes to biology, we seem to operate on a system of scientific freedom, and trust us, we're experts.
It begs the question, is the knowledge worth the risk? Is knowing if a virus can kill us worth the risk of creating the virus that kills us?
It's a question that feels totally abstract until you see a mouse with white eyes. In light of GXP2V, do we need a global pause? Do we need a biological non-proliferation treaty? Or do we just cross our fingers and hope the BSL-2 fan is working? But I want to leave you with one final thought, something we haven't touched on yet.
What's that?
We've been talking about massive labs and state-run militaries, but the technology to sequence and print DNA is getting cheaper and smaller every year. What happens when the digital code for a virus like GXP2V isn't just sitting on a preprint server, but gets downloaded to a desktop DNA synthesizer in someone's garage? The arms race might not just be between nations anymore. It might be coming to a basement near you.
That is an entirely different level of terrifying.
On that note, don't have nightmares, but do pay attention. Thanks for joining us for this deep dive.
Stay curious and stay safe.